Aspirin is a very common medicine for the treatment of pain. We use it alongside things like acetaminophen or Tylenol or ibuprofen, Advil, to treat common ailments, to reduce fevers. One of the issues with aspirin, however, is that it can upset your stomach. In young children, it can cause Ray's syndrome, whereas other pain relievers don't have that particular issue. Traditionally, pain relief came not necessarily from acetylsalicylic acid, which we know as aspirin, but its precursor, salicylic acid. And this can be isolated from willow bark and another few plant organisms. One of the issues with salicylic acid by itself, and again, this is known in traditional medicine, is that it can cause significant gastrointestinal problems and poisoning at high doses. So one of the things that once salicylic acid was found, attempts were made to change its chemical structure by doing some organic reactions to make it less toxic, to have less gastrointestinal problems. And that's how acetylsalicylic acid was synthesized and begun to be used. You are going to do a synthesis of aspirin from willow bark via a Fischer esterification reaction. So the first reaction that was used to form acetylsalicylic acid was a reaction between salicylic acid, acetic acid, using some additional acid to do the Fischer esterification reaction. The product of the Fischer esterification reaction from an alcohol and a carboxylic acid is an ester and then one molecule of water. This is very similar to the formation of isopentyl acetate that you will also do this week. So another Fischer esterification reaction. One of the important tests that you can do in the formation of aspirin is an iron 3 chloride or ferric chloride test. This tests for the presence or absence of phenols. So salicylic acid is a phenol. It is an alcohol that is attached to an aromatic ring. So when you react a phenol with the iron 3 chloride solution, it can turn purple or blue or green or some combination of those colors. If you successfully form the acetylsalicylic acid, that phenol will be gone and you should not see any color when you run the iron 3 chloride test. So you can directly compare that test via the starting material, salicylic acid, and the product, acetylsalicylic acid, and see the difference in the color. Now, one warning, as always, if you ever make anything that seems food-based or something that you could potentially take as a medicine, if you make it in the lab, you never take it yourself because you don't know the other components that could be within that mixture. So don't chew on the willow bark, don't try the product.